Good afternoon, everyone. Today, me and my classmates are talking to you about Chapter 5, starting with membrane proteins and their functions. Today, we will start talking about the two different types of membrane proteins, which are integral proteins and peripheral proteins. Integral proteins penetrate the hydrophobic interior of the lipid bilayer. Peripheral proteins are not embedded in the lipid bilayer at all. They are loosely bound to the surface of the membrane, often to exposure parts of integral proteins. Now we will start talking about the different functions of some membrane proteins, starting with
I am the transport. I transport the protein to the cell membrane.
Then next we will talk about
enzymatic activity.
In enzymatic activity, a protein is built into the membrane of an enzyme of its active site and exposed to synthesis in adjacent solution. Now, we will talk about
The signal transduction.
In signal transduction, a membrane protein or a receptor may have a binding site with a specific shape that fits the shape of the chemical messenger, such as a hormone. And now we will talk about
The cell recognition.
Some glycoproteins serve as identification tags that are specifically recognized by membrane proteins of other cells. This type of cell-to-cell binding is usually short-lived compared to shown as in the picture here. And now we will talk about
Intracellular joining.
In intracellular joining, membrane proteins of adjacent cells may hook together in various kinds of functions, such as gap junctions or tight junctions. This type of binding is more long-lasting than shown in this figure here. Then finally,
The extracellular matrix.
or also known as the ECM, are microfilaments or other elements of the cytoskeleton that may be non-covalently bound to membrane proteins, a function that helps maintain cell shape and stabilize the location of certain membrane proteins. And these are all functions of our various membrane proteins. Next, we will start talking about passive transport in a little more details.
There are three types of passive transport, and we have diffusion, facilitated diffusion, and osmosis.
Going further into osmosis, we can talk about the water balance of cells, starting with
Hypertonic solution, sugar.
in the picture you can see here, the hypertonic solution in the cell will lose water, shrivel, and probably die. Next, we will talk about
Isotonic solution. Normal.
if a cell without a cell wall, such as an animal cell, is immersed in the same environment that is isotonic to the cell, there will not be no net movement of water across the plasma membrane. And next, we will start to talk about
Hypotonic solution.
in a hypotonic solution, you can see as here it says it's lysed and as if the cell is bursting. Water will enter the cell faster than it leaves, and the cell will swell and lyse, like an overfilled water balloon. This is the water balance of living cells in their entirety, showing their similarities and differences. Good afternoon. Today, me and my classmates will begin our presentation by talking about Chapter 8. There are two stages of cell respiration and photosynthesis, one being light reaction and the other being the Calvin cycle. Inside of light reaction, it is the first step into changing light energy into chemical energy for the plant, which is essentially photosynthesis. Next is the Calvin cycle, or also known as the dark reactions. They are broken up into three different phases. The first phase being carbon fixation. Carbon fixation is when CO2 is attached to the RUBP molecule. After this, we go into the second stage known as reduction. Inside of reduction is the receiving of the phosphate group. The phosphate group is attracted to the other in this reduction phase. Next is the regeneration of RUBP. In this stage 3, the RUBP molecule that starts the cycle is regenerated so that the cycle can continue on. Me and my classmates will talk about the different stages of mitosis. Mitosis is separated into 6 different phases which help change one cell to form 2 cells. The beginning phase is called interphase. Interphase has 3 different stages. The first stage, G1, which is cell growth and organelle replication. The S phase, which is the DNA is arranged in pairs of identical chromosomes, and G2, prep for the mitotic stage. Next is the prophase. In prophase, chromosomes are visible, and the centrioles separate and move to opposite poles. After the prophase is the prometaphase. In the prometaphase, the kinetochore attaches to the microtubules, as seen here in the picture. After that, we move to the metaphase. In the metaphase, they all line up across the center, connected to the spindle fibers at the centrosome. Next, we move into the anaphase. Inside of the anaphase, chromatids separate into individual chromosomes and they pull apart, as you can see here by the separation of the chromosomes. Next is the telophase. Here, they gather at opposite ends and form around the spindle fiber, and they both disappear, which helps create 2 identical daughter cells, which we also call cytokinesis. Thank you for being with us today. We will go into Chapter 7, talking about aerobic respiration. The first part of aerobic respiration is glycolysis. Inside of glycolysis, it strips the electrons from the glucose to make 2 pyruvates. This happens in the cytosol. Next is transition, where they transfer the electrons to the mitochondria to the Krebs cycle in the mitochondrial matrix. The Krebs cycle makes 2 ATP, 4 NADH, and 2 FADH2. This all occurs inside of the mitochondria as well. The next stage is oxidative phosphorylation. In oxidative phosphorylation, it is separated into the electron transport chain, which is for ATP synthesis, and chemiosmosis. This helps the transport molecules in their inner membrane. It also happens inside of the inner membrane.
A lot of ATP. A lot.
Me and my classmates will talk about the overview of cell signaling. From the perspective of the cell receiving the message, cell signaling can be divided into 3 stages: signal reception, signal transduction, and cellular response. When reception occurs at the plasma membrane, the receptor shown here starts the process to send the transduction stage. It is usually a pathway of several steps, but here we have 2 of our classmates as 2 of the steps. The transduction relay molecules in the signal transduction pathway, with each specific relay molecule in the pathway bringing about change to the next molecule. The final molecule in the pathway triggers the cell's response. The response may be almost any imaginable cellular activity, such as catalysis by an enzyme, for example, glycogen phosphorylase. These all combined are a great example of cell signaling. Thank you for joining us.